Hi, my name is Beth Patel. I'm a development technologist in the clinical genomics lab at Mayo Clinic. I'm really excited to share a retrospective study with you today involving the utility of RNA-seq and mate-pair sequencing to collaboratively decipher structural variation. These methodologies are often seen as competing, but we've found that they often complement each other very well. First, I'm going to describe mate-pair sequencing and RNA-seq technologies themselves to give some background. Then I'll go into our retrospective study cohort, and then I'll run through two different cases that show synergy between these two methodologies. So the important thing to know about the mate-pair sequencing method is that the actual sequences that are resulted from this method are derived from a library prep that circularizes the DNA. Therefore, those reads should be about 2 to 5 kb apart in a specific orientation on the same chromosome. These fragments are called concordant fragments. When the reads do not map 2 to 5 kb apart on the same chromosome or to different chromosomes, these fragments are called discordant fragments and may indicate structural variation. The RNA-seq method, on the other hand, that was used for this study, used the Illumina TrueSeq RNA library prep kit and leveraged a poly-A capture system to identify expressed fusions. So I just wanted to give a brief comparison of these methods. Mate-pair sequencing has a base pair coverage that is very low, under 5x, but if you include the inferred sequence, the remainder of the circle, bioinformatically, we can add that into a calculation of bridge coverage, and that is generally over 30x. RNA-seq has a coverage of at least 20x at a fusion position. Mate-pair sequencing is a whole genome assay, whereas RNA-seq is clinically validated to analyze 571 genes. Some limitations of these assays: mate-pair sequencing will not identify structural variants with footprints below 30 kb, and repetitive DNA is a hurdle, as it is with many short-read sequencing assays. RNA-seq is not a whole transcriptome assay, and also RNA-seq cannot detect structural variants that don't result in a fusion. However, if you know of a structural variant on the DNA side, you can run a targeted interrogation for expression in that loci. In mate-pair sequencing, rearrangements are described as junctions, and in RNA-seq, they are described as fusions from exon to exon. In this study, we retrospectively collected data from all the samples that had both mate-pair sequencing and RNA sequencing performed over about a 2-year time frame. We had a total of 15 samples, including 7 solid tumor and 8 hematologic samples. 8 of these 15 cases were only fully characterized with information from both assays. 5 of these 8 cases had expressed fusions associated with a DNA rearrangement detectable by mate-pair sequencing. And in all 8 of these cases, we had informative expression results to guide the interpretation of the DNA rearrangement. And I should mention that the expression data were only analyzed as a part of this retrospective study. In our first case, we see a 63-year-old male with a benign lipotomous tumor and possible spindle cell sarcoma. The soft tissue is from the left deltoid, and this patient has a history of prostate adenocarcinoma and portal fibrosis of the liver. Additional genomic testing that was performed on this patient includes solid tumor chromosomal karyotype, which observed a translocation 5-6 in all of the cells. For this reason, we're wondering if this could potentially be from the germline, so we suggested a peripheral blood chromosome study to rule out a constitutional abnormality. However, no additional specimen was received. MDM2 FISH for amplification was also performed and was negative. So this is a mate pair sequencing genome plot. All the chromosomes are visualized horizontally on this graph. Chromosomes 1 through 12 and X are on the left, and 13 through 22 and Y are on the right. You can see C and V's color-coded red for lost and blue for gain along the genome, and structural variants are annotated with these blue circles, and black lines between each locus in the junction. The rearrangement we're interested in for this case is the translocation 5-6. This is called the junction plot, and this is how we look at structural variants in mate pair at a higher resolution than we're able to on the genome plot. And so based on the direction of the reads, which are color-coded, we can predict a 5' to 5' and 3' to 3' rearrangement product that will not result in a fusion. We can also predict 2 small deletions on each locus in between the breakpoints of these junctions. And there is 1 exon on 1 isoform of IL run. So based on the structural variant that we observed by mate pair sequencing, we interrogated the expression around both loci using RNAseq. As expected, no fusion was detected between these loci. EBF1 expression, as well as the genes around it, were normal compared with GTEx normal cultured fibroblasts. And we did notice underexpression or loss of expression of IL run, which was pretty interesting. We also see low expression of a couple other genes in this region, but this was the one that was intriguing to us since it was directly involved in the structural rearrangement. This case was particularly interesting because the information on the IL run gene was actually published after we had initially signed out this case clinically. And IL run is an inflammation and lipid regulator. It would be really interesting to see if the rearrangement is germline versus somatic in this case. I think that would tell us a lot about what's going on with this patient. And more studies are really needed to understand if there is truly a link between the loss of function of IL run and lipid-related tumorigenesis. Our second case is a heme case with a 51-year-old male diagnosed with de novo AML. Some other genomic testing that was performed on this patient included a chromosome study which observed trisomy 8 in 18 out of 20 cells. Our extensive FISH panel identified a NUP98 rearrangement in addition to trisomy 8. And for more information on NUP98 rearrangements, I hope you are able to tune in to Dr. Shinji Shu's presentation earlier in this meeting. Our NGS myeloid panel was also able to detect a FLT3 internal tandem duplication. This sample was included in a prospective mate pair sequencing study with RNAseq performed prospectively as well. So we didn't know what we were looking for when we had run the sample on both of these methods. On this genome plot, we can clearly see the trisomy 8 and the NSD1 NUP98 translocation. And it's not surprising we didn't see this by chromosome because the translocated pieces of chromosomes 5 and 11 are so small that this translocation is cryptic to chromosomes. What we're really interested in is this subclonal rearrangement involving chromosomes 1 and 10. So when we see complex structural variants, we like to try to reconstruct the derivative chromosomes based on the orientation of the reads we see by sequencing. So in this case, our derivative chromosome 1 will contain most of chromosome 1, a small inverted part of chromosome 1 that we see here, and then the 10p terminus and containing part of MLLT10. This region of MLLT10 is right next to a 5' region of USH2A. This is not predicted to create a fusion product because it's a 5' to 5' rearrangement. The derivative chromosome 10 will contain the entire 10q arm up to the middle of MLLT10, and that is adjacent to extragenic material. So we wanted to see, how does this disruption of MLLT10 affect expression? So RNAseq, of course, detected the NUP98 NSC1 fusion. And when we looked at the expression of MLLT10 compared with GTEx normal blood, we found that there was an overexpression. We also see overexpression of this adjacent DNA JC1, but that's pretty frequently overexpressed in blood and bone marrow, and we didn't deem this clinically relevant. So MLLT10 rearrangements are most commonly seen as fusions in AML with KMT2A and PCALM. Overexpression of MLLT10 is also documented in colorectal and lung cancers. And although this isn't the primary abnormality, we can interpret this to mean that the AML is progressing. So RNAseq clarifies the expression effect of the MLLT10 disruption that we see by mate pair sequencing. So in summary, we conclude that at least in this subset of cases, mate pair sequencing and RNAseq are synergistic. They can both detect structural variation, but we have to think of them in terms of DNA and RNA, structure and function, anatomy and physiology of omics. Often these two methodologies are seen as competing, but in our solid tumor samples, all of the samples that we run, we could use the expression data or the fusion data to clarify whether structural variants had a functional effect. With that, I'd like to thank all of the talented people at Mayo Clinic that make this work possible, and also the Cancer Genomics Consortium and you for listening today. So thank you.